No, tomorrow's lab is follow-up from Wednesday's lab. So nothing new tomorrow. Okay, hopefully we'll be able to finish up today the material that is the breakpoint for the exam. I suppose if I had to, I could take a couple minutes on Friday because there's nothing new Friday, but let's see what we can get covered. So we're talking now about chemical inhibition of microbial growth. Remember that these substances typically are intended to be used for either as disinfectants or antiseptics, kind of depending upon their toxicity to tissue. And we were in the process of talking about the phenolic compounds, and specifically the bisphenolic compounds, which are two phenolic rings joined together. In the middle, we have that orthophenylphenol, very simple bisphenol, which is one of the ingredients in the cleanser Lysol. And at the end of the period, I said that one up at the top, the hexachlorophene, is an interesting molecule that we should talk about just a little bit. This particular bisphenol used to be used a lot in acne treatments. There was a substance called Bisahex that was the rage when I was young for acne medication that contained a lot of this hexachlorophene. And it also used to be very common to bathe newborn infants before they left in the hospital in a hexachlorophene solution to try to prevent them from bringing home hospital-acquired infections, particularly like staph. And it worked pretty well. The problem is that later investigations found out that hexachlorophene could be absorbed through the skin and get into the nervous system, and particularly in young developing infants, cause neurological damage. And there was some real serious damage to a lot of infants because they just didn't realize that this was particularly the case. So the use of hexachlorophene was quickly discontinued with a couple of just big exceptions, and you can't get hexachlorophene in anything without prescriptions anymore. It's been phased out in favor of the bottom bisphenol, chlorhexidine, and chlorhexidine doesn't have that neurological problem. It's still an effective antibacterial agent. It is the agent of choice, at least right now, for scrubs to try to prevent acquiring MRSA. And so chlorhexidine is still a very effective and pretty safe antibacterial bisphenolic agent. The one that's hand-drawn in up at the top up here, triclosan, has been in the news recently. Does anybody recognize triclosan or heard of it before? Triclosan is typically the active ingredient in any soap that says that it is antibacterial. So it is a phenolic, bisphenolic compound as well. The reason that triclosan is in the news right now is that the FDA has done long-term studies of antibacterial soaps and has determined over those long-term studies that antibacterial soaps are no more effective than normal soaps. Now, there's some question about why that's the case. One idea is that bacteria have quickly evolved resistance to this particular compound, triclosan. But the FDA has mandated that manufacturers start phasing out triclosan and no longer put it in soaps because they have determined that the potential health risks of this added chemical don't merit, inclusion in the soap, because the soap really isn't that much more effective with triclosan than without. So you're going to see that being phased out. That particular ban does not affect any of the hand sanitizers or liquid alcohol gel compounds. Those were not affected by that particular ban. So those are some of the phenolic compounds of interest. There are a number of antibacterial compounds that fall under the category of being surfactants. And surfactants typically work by affecting cell membranes. One of the things that surfactants can do is they can just kind of solubilize the cell membrane and kind of strip it away. But more commonly, the mode of action of surfactant molecules is that they insert themselves into cell membranes. They are depicted up here as these little squares with sticks on them. And those surfactant molecules interact with the phospholipid bilayer and insert themselves into the membranes and create artificial channels which make the membranes more leaky, and things enter and leave the cells that shouldn't. And so they interfere with the permeability properties of the membrane. There are two major compounds, or classes of compounds, that are surfactants, soaps and detergents. One of the major differences in soaps and detergents is that soaps tend to be anionic compounds and carry a net negative charge, and detergents tend to be cationic compounds and carry a net positive charge. Typically, soaps are long hydrocarbon chains. This is showing it in the sodium salt form, and that's why there's a positive charge here. But the anionic portion of the soap is this long hydrocarbon chain with a couple of oxygens at the end. Soaps are mildly effective as antibacterial agents because of their surfactant characteristics and inserting into the cell membrane. But the main utility of soaps is just physical removal of bacteria. And so they typically are important strategies in the process of degermination. Remember, degermination involves scrubbing and also using agents that help to facilitate the removal of oils, for example, from your skin, and soaps will do that. But in terms of killing bacteria, soaps aren't so great. As a matter of fact, if you took a culture of a soap dish, it probably wouldn't be uncommon at all to find several species of bacteria actively growing in that soap. Now, there are some things you can add to soap, such as alcohol, which will then enhance the killing ability of that soap. Soap to which alcohol is added is sometimes referred to as green soap. On the other hand, the detergents, also which contain large hydrocarbon chains, contain some kind of a positively charged molecule at the end of that hydrocarbon chain. The major type of detergents that we're going to refer to are a group of compounds called quats. Quat stands for quaternary ammonium compound. And so this is a quat right here. Acetylpyridinium chloride. Does that name ring a bell at all? Back to freshman year. Right now, over in the 134 lab, they are doing the mouthwash lab. You remember that one where you put the little filter paper discs? What did most of you hypothesize, do you remember, was going to be the most effective mouthwash that we were going to use? Most people hypothesized Listerine because, you remember why? It had the most alcohol. And as it turns out, in our experiment at least, Listerine was one of the least effective mouthwashes. Other mouthwashes worked better, and when you went to that list of ingredients and started to try to determine what did they have in common, what did the ones that worked well have in common, and what did the ones that didn't work well lack? Well, one of the ingredients that we tended to narrow down on was this compound, acetylpyridinium chloride. Trade name is Cipra. That was found in Scope, for example, which was one of the effective mouthwashes. So that quaternary ammonium compound, which is really a detergent, was really the active ingredient in many of those mouthwashes that worked well. It's pretty effective. Now, the question is are alcohols antibacterial? And the answer is yes, they are. Not all alcohols have the same killing power, but all of them are pretty effective in denaturing protein. Alcohol also has a tendency to precipitate another important cellular compound. Anybody know what it is? In the presence of alcohol, this compound settles out of the cytoplasm and forms this gooey mass of material that we can then separate out from the lysed solution by spinning it out on a glass rod. Have you ever done that in class or seen that observation? This is the dirty, easy method to isolate DNA from a cell. Okay, alcohol precipitation, and then twirl it out on a glass rod and get the stuff that looks like snot, which is the DNA, and it's alcohol precipitated. Okay, so alcohol does a couple of important things. It can denature proteins. It can precipitate DNA. The alcohol that's most effective in being antibacterial is ethanol and also isopropyl alcohol. Isopropyl alcohol is sold in drugstores as rubbing alcohol, and it has some decent antibacterial properties. Some alcohols are not as efficient. Methanol is a pretty poor antibacterial agent, so it depends upon the nature of the alcohol whether it works well or not. When we first started talking about chemical compounds, I told you that there was kind of a rule of thumb that in general, things are more effective in killing at the higher concentration you have it, but that there were exceptions to that rule. Alcohol is one of those exceptions to that rule of more is better. So if you compare a solution of 100% ethanol to a solution of 70% ethanol or a 70% isopropyl alcohol solution, this one's going to be better. A lower concentration is actually desirable. How come? Because some of that water that is present in the 70% solution, it's 70% alcohol, 30% water, is necessary to help facilitate the denaturation of the proteins. Remember we said the proteins tend to denature better in the presence of water? Another factor is that 100% alcohol solutions tend to dehydrate tissues and pull the water out of them. And remember, in the absence of water, denaturation is less effective. So 100% ethanol is oftentimes used now as a preservative for tissue specimens. We used to use formaldehyde. Anybody knows why we don't use formaldehyde as much anymore for preservation? Carcinogenic. Okay. There's a lot of old comparative anatomists out there who spent their lifetimes dissecting formaldehyde-embedded specimens, and many of them have died from brain cancer. Okay. Including the guy that I had studied comparative anatomy. So, why didn't Listerine work very well in our experiment? Alcohol is antibacterial, but why didn't Listerine work very well? Well, one of the limitations of alcohols is that they tend to evaporate fairly readily. And if you think about it, you take these filter paper discs impregnated with Listerine, and you seed them onto a plate of bacteria, and then what did we do? We put those plates in an incubator, and the incubator was 37 degrees Celsius. That's pretty warm. So probably what was happening is most of the alcohol was evaporating out of the disc before it had a chance to diffuse out into the media and kill the bacteria. So it was really a product of our experimental design more than it was a reflection of the fact that the alcohol didn't have very good killing power. Some of the most effective antibacterial agents are the halogens. Halogen compounds include chlorine, bromine, fluorine, halogen compounds work by being strong oxidizing agents. And particularly what they tend to do is they tend to break disulfide bonds in proteins. They can also oxidize, remove the hydrogen from sulfhydryl groups. And this can have a real dramatic effect on the tertiary structure of proteins and basically contributes to their denaturation. By far, the halogen that has the most commercial applications is chlorine. You'll occasionally see bromine used. Bromine's a little bit harder to control. It's a little bit more toxic. Hot tubs, for example, oftentimes will use brominating tablets as opposed to chlorine tablets for disinfection. Fluoride toothpastes have some antibacterial properties. Fluorine has some inhibitory effect on the bacteria that adhere to your teeth. But primarily the halogen of interest is chlorine.
What about iodine?
Iodine is considered to be a heavy metal and not a halogen. So we'll talk about iodine in a different group. Okay? Iodine, mercury, copper, silver are kind of a different category. Yes.
They do have anti-bacterial. Iodine does have properties though, doesn't it?
Yes, it does.
So
Yeah.
you might put it in Greek water.
Mm-hmm. Absolutely. Yeah, we're just going to put it in a different category of compounds. The three major forms of chlorine that are used in disinfection are hypochlorite. Chlorine gas is sometimes used, typically for larger scale water disinfection. And a compound called chloramine. When you add any of these forms of chlorine to water, they interact to form hypochlorous acid. And it's the hypochlorous acid that is actually the oxidizing agent. Hypochlorite you are probably familiar with in another context. This is bleach. And bleach solutions are used routinely to disinfect things. Pretty effective. One of the downsides of chlorine, particularly of chlorine gas, is that chlorine gas tends to react with organic matter. And one thing that happens is it can become chelated and therefore inactivated. But something more insidious happens oftentimes. When chlorine interacts with organic matter, a class of organic compounds called trihalomethanes are oftentimes produced. And trihalomethanes are nasty little buggers. They are carcinogenic. Well, think about the implications of that for a second. One of the primary ways that we do final disinfection of water after it goes through sewage treatment is gaseous chlorination. Typically during sewage treatment, bacteria facilitate the breakdown of organic matter, fecal material, and convert it into inorganic substances, but they don't get it all. And so there still can be organic matter in the final water that is chlorinated. And what happens? Exposed to chlorine gas, trihalomethanes can form. And so there are detectable levels of trihalomethanes in water coming out of sewage treatment plants. Now, the first response might be, well, let's stop chlorinating. Well, think about the public health threat of water that's not chlorinated and getting exposed to all those pathogens. There's a risk with everything, and it's been determined that the public health risk is greater from consuming unchlorinated water than the cancer risk from trihalomethanes. But is there an alternative? Well, we talked about another potential alternative, ultraviolet radiation. But that involves a massive just re-engineering of a water treatment plant. Another possibility is to use not chlorine gas, but another form of chlorine, chloramines. And the advantage of chloramines is they don't have this particular reaction. They don't form those trihalomethanes. Unfortunately, chloramines are really expensive compared to chlorine gas. And so there is a financial reason. Okay, you're right. I should have put iodine here. It's not heavy metal. It's another halogen. So what about iodine? Does iodine have antibacterial properties? Yeah, it's a halogen as well. Iodine can irreversibly bind to sulfhydryl groups like chlorine does, so it has this very similar mode of action, and denature proteins. It tends to cause irreversible denaturation. Once it binds, a covalent bond forms, and it's basically stuck on that enzyme or protein and permanently alters its shape. The process of iodine binding to other molecules is called iodination. And so iodinating proteins is a pretty effective way to denature them. You ever had iodine put on a cut? What's the response? Iodine shouldn't bubble, peroxide will. Did it hurt?
Yeah, but it made like a pasty, not like a pasty, but like gunky stuff on top of it.
Okay, kind of a skinny kind of thing. But oftentimes iodine solutions also hurt. Iodine is typically sold over the counter as a tincture. Do you remember what a tincture was? It's not a water solution, it's an alcohol solution, okay? And that halogen iodine tends to irritate nerve endings and cause pain. However, if you combine iodine with a surfactant, that combination is called an iodophore, you lose that irritating property of iodine, and it's still pretty effective at killing microorganisms. You ever seen the stuff, if you've observed the surgery, where they squirt some brown liquid on an incision before they make the incision? That's an iodophore. That's betadine, probably. There's another one called Ovapone or something like that. But these are iodine surfactant combinations, and they are less irritating to nerve endings than plain old iodine is. And that is right. You can also use iodine for emergency disinfection of water. If you have one of those little bottles of tincture of iodine, you can still probably buy in drugstores. If you put 5 drops of that in a quart of water for about 20 to 30 minutes, it will disinfect the water. It doesn't taste real great. It's got that iodine taste to it, but at least you have killed the non-spore forming pathogenic microorganisms. Heavy metals. When we first started this section, I said that Sometimes we've known that particular methods of treatment are effective long before we've understood the mechanism of how they actually worked. And heavy metals are a good example of that. Substances like copper, gold, mercury, silver have long been identified as things that kill microorganisms pretty effectively. So even back in the Roman Empire, rich people drank out of cups not made of wood like the poor people did, but of cups of silver and gold. Yeah, there was a status because they were precious metals, but there was also a health benefit, okay? Because those particular materials in the drinking vessel tended to have antibacterial properties, particularly in even very small amounts. Substances that are inhibitory in even very, very small amounts are said to exhibit oligodynamic action. And that's clearly illustrated up here on this particular seeded plate with bacteria. This is kind of a variation of the mouthwash experiment shown here. But instead of putting filter paper discs impregnated with antibiotic or mouthwashes, here little pieces of metallic compounds are put on the surface of the plate. Here's some little silver amalgam from a filling, for example. Here's a little piece of gold foil. And these clearly have inhibitory properties. You can even put coins that contain high metal content on a seeded agar plate and see some oligodynamic action. Copper is primarily effective against autotrophic organisms by inhibiting photosynthesis. Copper ions tend to inhibit photosynthesis. Silver still has a number of applications in medical settings. I think I mentioned this before when we talked about chlamydia, but when newborn infants were released from the hospital, oftentimes they were given preventative or prophylactic eye drops in case the mother had an undiagnosed chlamydial infection, because going through the birth canal you can pick up those bacteria and cause horrible eye infections. They used to use silver nitrate drops, but they were pretty painful. Those typically have been replaced with antibiotic drops now. But silver nitrate is a pretty effective antibacterial agent. Works much the same way that the halogen compounds do. These heavy metals tend to complex with proteins irreversibly. And especially if they bind at their active site, they can denature the protein and make it non-functional. A lot of the heavy metals are quite toxic. I don't have cadmium on the list, but cadmium is a very toxic heavy metal. Catheters are one of the medical devices that tend to be of great concern in terms of introducing contaminants into the body. And so catheters are oftentimes coated with actually pure silver, and that silver has a very antibacterial inhibiting property. And so the use of silver in association with catheters is pretty common. Mercury is an interesting antibacterial agent. Unfortunately, the more we learn about mercury, the more we appreciate its inherent level of toxicity. When I was little, playing with mercury was a great diversion. Mercury has some interesting properties. Okay? And if a thermometer broke, what did you do? You played with the mercury. Well, inhaling mercury vapors, we found out, isn't great. And that's one of the reasons why over time, we have, and most labs have, discontinued mercury thermometers in place of alcohol thermometers. So the toxicity of mercury is becoming increasingly appreciated. There used to be something that was kind of the equivalent of tincture of iodine you could buy at the drugstore. There were little bottles of mercury compounds, merthiolate, and mercurochrome. I haven't looked recently, but I'm not sure they even sell these anymore. Okay. They didn't sting like iodine did, but as it turns out, the toxicity was something that probably we shouldn't have been messing with. Probably the form of mercury that is attracting the most attention within the last decade is an organic compound called thimerosal. Have you heard of thimerosal before? It's a preservative that used to be used fairly routinely in vaccines. And this is the origin of the controversy, particularly about what has now been discredited, the association between autism and vaccines. And people believe that it was the thimerosal preservative that was the culprit. Thimerosal has been discontinued as a preservative in most vaccines. There still are a couple of vaccines that use thimerosal preservative because other preservatives have not been effective. Some forms of influenza vaccine, although I believe everything that they are doing over in the health center for flu vaccines is thimerosal free. You could ask Kathy over there if you're going to get your flu vaccine, which you should. And there are some TB vaccines that are also still using thimerosal preservative. But we're really discontinuing that, even though the original research that was done associating the vaccines and autism were really shown to be invalid. Another class of compounds that have antibacterial properties are just plain old acid bases. Ammonium hydroxide is an active ingredient in many floor cleansers, for example. And by obviously changing the pH, that can have inhibitory effects on the bacteria. Acidic compounds are used a lot in preservatives of food. One of the first acidic compounds that was used widely in food preservation was acetic acid. Pickling, okay? Pickled cucumbers, pickled cabbage, okay? A lot of organic preservatives that are acidic have also been developed over time. And if you look at the list of ingredients of food on food labels, many times they're loaded with organic acidic preservatives. Particularly ones that you might have heard of before include benzoic acid. It's a pretty common food preservative. Again, reasonably effective at retarding bacterial growth, but there are health concerns associated with ingesting some of these kind of compounds with your food supply. And that concern has manifested itself in a real resurgence in people interested in having food that's preservative-free, organic food that is not treated in that particular way, also isn't treated with pesticides or herbicides or things like that. So consumers are really becoming much more savvy about what they put into their bodies and are becoming much more concerned about what are some of the agents that are being used to try to retard microbial growth. Aldehydes. These particular molecules interact with proteins through a process called alkylation. So they actually physically bind to amino acids on the proteins. And during the process of alkylation, proteins become cross-linked to one another. They kind of glom together. So over on the left over here, this is one of the aldehyde compounds we'll talk about in a second called glutaraldehyde. And that binds to particular amino acids on membrane proteins here. This would be in the LPS layer. And it causes those proteins to become cross-linked to one another. Aldehydes can also interact with DNA and cause cross-linking of DNA molecules. So they kind of polymerize, they kind of glom together. And that tends to make them non-functional. One type of aldehyde that's pretty effective, and actually is so effective that under the right conditions, it can cause chemical sterilization. It not only kills vegetative cells, it also kills endospores. Glutaraldehyde is a liquid. It's kind of a yellowish, sticky liquid. And one of the things that you can do to treat things like surgical instruments is you can just soak them in tubs of this compound, glutaraldehyde. For large-scale treatments, glutaraldehyde is used in a device called a chemiclave. So over here is a chemiclave on the left, and this is a device that kind of bathes whatever you're treating in a glutaraldehyde solution. Takes quite a while. Typical treatment takes, hours, 2 or 3 hours at least, to achieve chemical sterilization. Far less than autoclave treatment would. So when might you have to use this compound as opposed to autoclaving? That's a limitation of autoclaving for some things. They're heat sensitive, right? So heat sensitive materials oftentimes can't be autoclaved. A gaseous form of aldehyde is ethylene oxide. So this is a liquid. Ethylene oxide is also cross-links proteins, cross-links nucleic acids, and is also very good for treating heat-sensitive material, particularly plastic material. Did you ever wonder how those sleeves of Petri dishes that we're using all the time get sterilized? We can't autoclave them, can we? They would just all melt into a bundle. They probably went through a very large device that exposed them to fumes of this ethylene oxide. That particular device is shown up on the right. That's called a gas autoclave. There are a couple of downsides of ethylene oxide. And one is that it's pretty toxic. It's probably carcinogenic to humans too. Ethylene oxide also tends to interact with DNA. It has a high specificity for the guanine nucleotides. It tends to alkylate those guanines and cross-link DNA molecules. So a gas autoclave is going to have to be set up in such a way that the user is not exposed to those particular fumes. But we can do that. We can engineer that. Another problem with ethylene oxide, though, is that it's explosive. That could be a problem. However, if you mix ethylene oxide with carbon dioxide, it's no longer explosive. So you'll see on this particular little diagram over here, one of the things that's part of this gas autoclave is an ethylene oxide cylinder, but also a CO2 cylinder, and those two are mixed together before they're introduced into the chamber. The CO2 doesn't interfere with the ability of the ethylene oxide to be antibacterial, but it does keep it from being explosive. And then last category, are a group of compounds called oxidizing agents. The major one we'll talk about is hydrogen peroxide. And we've already talked about how hydrogen peroxide tends to be antibacterial. Remember that hydrogen peroxide itself is an ROS. But remember also that when peroxide is detoxified by things like the peroxidase enzyme, it can also generate its own ROSs. To be really effective, though, hydrogen peroxide has got to be in a very high concentration. Here's a device that's really important for treating an expensive but important medical instrument, an endoscope. Okay. After someone has a colonoscopy, obviously that endoscope has to be sterilized before it can be used again. And we can't autoclave it. Okay. This particular device that's used for sterilizing endoscopes uses high concentrations of hydrogen peroxide, up to 35%, which is really hot. Typical hydrogen peroxide that you buy in a grocery store or a drugstore for surface disinfection is about 3%. So very, very high concentrations of these. As it turns out, hydrogen peroxide that you buy at the grocery store is only marginally effective. Those bottles of 3% H2O2, not a very good disinfectant at all. And what's really misleading is that people look at the peroxide, and what's it tend to do when you put it on a cup? Bubbles, right? Why is it bubbling? Because peroxidase enzymes are breaking down the hydrogen peroxide. But people look at it and go, oh, it's bubbling, it's working. Actually, damaged tissue releases peroxidase enzymes. Your blood contains both catalase and peroxidase enzymes. And so when you pour that on a cut and you see the bubbling, basically you're seeing your body breaking down what's supposed to be the disinfectant. So it really doesn't work very well at all. It's not a high enough concentration to be that effective. Obviously, if any organisms are going to be affected, it would be anaerobic organisms, because remember, anaerobic organisms do not contain the proper enzymes to detoxify peroxides. Another oxidizing agent that has found some utilization more recently is ozone. And we tend to think of ozone in two different contexts besides being antibacterial. One context is a protective ozone layer, which filters out short wavelength EM radiation like gamma rays, X-rays, UV rays, very protective. Most people, when they think ozone, think automobile pollutant. Okay? But in fairly high concentrations in enclosed environments, ozone is also a pretty good oxidizing agent. It generates ROS species as well. And there have been some investigations into trying to use ozone as an alternative to chlorine in municipal water treatment. Hasn't really caught on that well, to be honest with you. And really, probably the only time that ozone would make sense is if you were building a new water treatment facility from scratch and you were able to engineer it using ozone technology. But to retrofit an existing water treatment plant to use ozone probably would be prohibitively expensive. This is just a smattering of the types of chemical compounds that can be used to control the growth of microorganisms. One of the things that I want you to appreciate is the fact that very few of these would be considered to be ideal germicidal compounds. They've all got some limitations. I said at the beginning, chlorhexidine, which is that bisphenol, and high concentration ozone, probably most approach ideal qualities, but everything else has limitations, and I've tried to highlight what some of those limitations are. All of these compounds are designed to be used as either disinfectants, inanimate surfaces, or as antiseptics, surface application of living tissues, but are not intended to be taken internally. You know, we fudge that with alcohol a little bit, but that's also toxic chemical. What we really need to do to get a real handle on disease, especially once the organisms have invaded the body, is something that we can take internally. And internal agents have to have selective toxicity. They can't be just this nuclear bomb that kills everything, because then the drug would kill you as well as the bacteria. So this idea of selective toxicity is going to be really important for things we take internally. Things we take internally are designated as being chemotherapeutic agents. So that's where we're going to go after this exam, is we're going to talk about some important chemotherapeutic agents, and particularly concentrate on antibiotics and all of the benefits of antibiotics and some of the health concerns that are associated with antibiotics as well. So tomorrow is a day to follow up on your plates that you did and other things that you might need to catch up on. So this is ending point for exam, okay?
So is this what you wanted for like the unknown that you check over? Is that the
Now wait, for what unknown?
I have 7.
All I need right now is I need gram reaction and cell morphology.
Okay, is that the morphology though? Like that info?
Oh, no, are the cells rods or are they coccytes?
Oh.
Okay, not colony morphology, cell morphology.
Okay.